Yes, so welcome to the day 9 session on molecular talking. Uh, is my presentation visible to you? Yes or no? Yes, whether the slides are moving?
Yes, sir.
Yes, it's moving, isn't it? Some kind of internet frequency. I'm just checking. Yes, it's moving, isn't it?
Yes, sir.
Okay, now let me go ahead. So before stepping on to today's session, can we go across the recapping of last session topics? Yes, come with the last session topics. So what exactly did we discuss in the last session? And today we'll be discussing extensively on molecular talking. Yeah, so last session we discussed on ligand-based approach, like we discussed on types of QSPR, that is what is QSCR, what is QSTR, what is QSCR, and what, so we have seen even related to electro QSER. Okay, so these are the types of QSPR we have discussed. And for biotechnology, we basically use QSER. QSAR stands for quantitative structure activity relationships. Okay, so and if you see this molecular descriptors, so we have types of molecular descriptors. The more majorly we find electronic descriptors for estimation of the properties of the ligands. If you want to check the activity of the ligand, obviously we use the correlation of physicochemical properties. The activity is determined as the function of physicochemical properties of the ligand. So, what properties we have chosen to determine the properties? And in computational, for a combination of the properties, we are describing through molecular descriptors. Okay, and based on the selection of molecular descriptors, you can see the value will be varying. Can be bioactivity, bioavailability, whatever correlations we are calculating, that will vary based on the descriptors, how it is correlated to calculate. And then we have discussed something on the types of models of QSCR. Basically, we can classify it as classification model and regression model. We have discussed in regression model, like we have a single linear expression, multiple linear regression model. And then we have discussed something on the terminologies like what is training data set and what is test data set. So we have discussed on there are two types of data sets available in QSCS studies. Later, we have gone to the see what if one participant is answering from beginning. How about the others? Other than QSR, what else did we discuss? You remember? I have shown you some tools for demo. What are the tools I brought for demo? Yes, see, the first half an hour session you are active. The next half an hour, I don't think. Change automatic picture in picture setting. What happened? I don't know, something is happened at present. Picture in picture is open because you are screen sharing. Okay. So tell me, what exactly did you discuss? Paddle, okay, cake, about the metabolic database. Did I discuss this on yesterday? What else did we? E-Dragon. Yes, we have discussed on these tools. These tools are a paddle, E-Dragon. These are used for QACR. Very good. Madam came across with a tool called MATLAB. You know, we have discussed a program in MATLAB for pharmacokinetic studies. Do you remember? What is the program in MATLAB we use for studying pharmacokinetic and pharmacodynamic models? We discussed on compartment models, non-compartment models, and in compartment models, we discussed on what is one compartment model, two compartment model, three compartment model, and then I showed you a model for breast cancer, and then I told you if the paclitaxel is taken over a period of 40 days in a particular dosage, after 7 days, what is going to happen. So all these things we have simulated in the MATLAB. Okay, whatever compartment models we built in the pharmaco related to pharmacokinetic we built in the MATLAB, we have executed the simulation after the modeling, isn't it? So now tell me, it comes under what is the tool, what is a program in MATLAB used for doing the modeling and simulation of pharmacokinetics as well as pharmacodynamic studies? See, only one participant is answering from the beginning. Your grasping power is reducing drastically. So, do your assignments on time. So, we are also. I'm also sending you the recorded videos, isn't it? Watch the videos. Biology is the tool. We have tools like bioinformatics tool to work on sequence alignment and structure prediction, etc. And biology is generally used for pharmacokinetic and pharmacodynamic studies. Of course, these are one of the important studies we have to consider as an important studies in drug design, isn't it? Now, after this QSCR, after biology, I also taken one more study, one yesterday. What is the study? I also taken you to the tool, an online tool demonstration. What is the online tool I've provided demo on yesterday? And what is the study? What is the method of that computer-aided drug design? If QSR is one of the method, what is the other method we have discussed? So this is what we call it a self-assessment. So we have discussed on pharmacophore modeling. Okay, mapping. You remember? This is the method we have discussed. And here we have discussed something called form mapper. You remember? Form mapper we have seen. And for charm, we have also seen probish charming. Probish charm. Okay, so they're using this, we have seen the results for whatever we worked on the earlier session. Okay, form mapper is the tool. Exactly. Okay, so this is used for pharmacophore modeling and mapping. Now let me take you to the molecular docking. There are much more concepts in this computer-aided drug design. What is molecular docking? And we discuss on ligand protein docking softwares, protein docking softwares, theory behind, and the scoring methods, which is most important to determine to distinguish the freewares from the commercial softwares of docking. Then we see some hands-on experience in Autodoc. Molecular docking. What is molecular docking? It is the computational procedure to predict the conformation of a protein ligand complex. I told you in molecular docking, we have two terminologies. One is ligand and another is the receptor. So the ligand and receptor can bind with each other to form a ligand receptor complex. So according to the principles, this is actually a reversible reaction. So the rate constant K1 and K minus 1 given for forward and reverse reaction. If this is a general concept, and if you consider in enzymology studies, enzyme can react with the substrate. It can give you the enzyme substrate complex, and this enzyme substrate complex under irreversible reaction, which can able to produce you enzyme and product. Why I am giving in square bracket? It means that particular concentration of ligand and receptor, concentration of ligand and particular concentration of a receptor, okay, is going to undergo the reaction and forms this kind of a complex. Okay, in case of enzyme substrate, this is the intermediate complex, and these are the final products you can find it going to form. Any molecular talking protocol consists of two components. One is search algorithm, the other is the scoring function. This only distinguishes the freeware from the commercial software. Many of them heard about Autodoc. Autodoc is a freeware tool supplied by MGL tools. Okay, so Autodoc is a freeware tools. Is a freeware software from MGL tools. And if you see, this Autodoc is going to give you only one kind of an output parameter called as binding energy. The binding energy is given in terms of kilocalorie per mole. So if this is something about Autodoc, if you see some commercial softwares, okay, even in this in a SVC, we have a commercial software called Discovery Studio. So Discovery Studio earlier we have procured from Aslaris. The name of the company is Aslaris. So this company has stopped providing this tool because they have sold it to another company called BioVia. Actually, Azaris is from USA and BioVia is from the Japan. So the BioVia Discovery Studio, if you see the scoring function, we have eight scoring functions. We have eight scoring functions. So each and every scoring function is a correlation of the physicochemical properties. Like if you see the physicochemical properties of the drug, the physicochemical properties can be a pKa, pH, and the size IC50, whatever properties can be molecular weight, and whatever is seen even in the Lipin scale rule, number of hydrogen bond donors, okay, number of hydrogen bond acceptors, donors, acceptors, okay, everything. Okay, we use a combination. One score is a combination of pK and molecular weight. The second function might be a combination of pK and hydrogen bond donor. Like this, we have a combination of parameters to calculate 8 different scoring functions. The objective of using, the price of this Discovery Studio at that time in 2009, okay, we procured a partial package. We didn't procure entire drug design software. We procured a partial thing. At that time itself, it was around 18.5 lakhs. The total package is around 25 lakhs. But what is the price of Autodoc? Even at that time, the price is zero. Okay, the price is $0. But here it comes around 25 lakh rupees. Okay, so only thing is that the difference you find from the commercial and freeware is here you find multiple scoring functions, and we find a few more tools. Like if you see energy minimization, we have multiple energy minimization tools in Discovery Studio. So we can use the energy minimization tool. We can find the scores in different forms based on the parameters. Like this, there are only the search algorithm and the scoring function can distinguish us one tool from the another tool. You got it? This is something about molecular talking. So after molecular talking, you can find this as an output. This is a output of a ligand. This is how a ligand looks like. The ligand is presently present in the form of stick model. So the ligand in stick model you can able to see, and this is the protein receptor in surface model. The protein is present in the form of surface. If you see here, the ligand is visible at present because the ligand is binding only on the surface. If the ligand is visible in the surface model, then we can tell this ligand as hydrophilic ligand because this is binding in the hydrophilic binding sites. Okay, so there are two types of binding sites. One is hydrophilic. The hydrophilic you can find only on the surfaces of protein. But if the ligand is buried inside, even if you scroll in all the directions, if you can't able to see the ligand, and if it is buried inside the sites, the buried site is what we call it as hydrophobic site. Then the word ligand, we can tell it is a lipophilic ligand. You remember when we discussed something on ADME, we discussed on BBB. What is that BBB? Blood brain barrier, which is more lipophilic, which is more lipophilic. It means that if you want to design a drug which can penetrate up to the brain, you should have a hydrophobic kind of a characteristics. Now let me take you to the protein ligand talking. There are plenty of tools. In 2000, I believe I used these kind of a tools. At that time, we use this kind of a uses of talk, SGI, okay, Silicon Graphics Interface. That is the operating system we used. So we installed this in HG and we started working it. It works for almost like a Linux, not Linux, Unix. In Unix, you don't find any kind of a uh, graphical user kind of a thing. We have to use only the keyboard events. But nowadays, you can see the docking softwares are well developed and it's more user friendly, especially if you try to do even with Autodoc, you feel it's more user friendly. But we have enormous steps in a tool called Autodoc, which is a freeware I was explaining from the beginning. You have plenty of steps and sub-steps. If you fail in any of the sub-step, you won't get the result. So you should be very careful in executing the protocol in Autodoc. But there are some tools, as I earlier mentioned, Discovery Studio. Okay, this is a package actually, not only used for simply docking, we can use it for multiple purposes. We can use it for sequencer, and we can use it for QACR, we can use for pharmacophore modeling, we can use for simulation, whatever we are discussing related to drug design. Okay, all the tools, all the programs are available in this single package. Okay, this is package, not like a tool like an Autodoc. So if you see this, this also use, and even in the Discovery Studio, we also have protein docking server. Everything is there. Okay, but if you see these tools, whatever I showed, this can be used only for ligand protein docking. The ligand here I am meaning that we use small molecules as a ligand, only the small molecules. So protein docking, both are macromolecules, isn't it? Protein talking, the best example is antigen antibody. So if you go want a humoral immunity, okay, there are two types of immunity, humoral and cellular immunity. For the humoral immunity, you obviously have to check these kind of affinity, immunity, obviously. Okay, for checking the affinity, obviously we go with the protein docking servers. Okay, we have even a scroid incher. You remember? This is one of the package, many uh, repeated institutes have this commercial package. This is still a package like a Discovery Studio. We have plenty of programs or tools to work with, and we also have a tool for protein ligand docking. Protein docking. So these are the tools, and most of the tools, whatever I have showed, okay, we can be executed in online. Okay, that's we have a client server. Just like that, you have to input two different protein molecules, and then you can find the protein interaction. Okay, mostly even this we can see the results in the form of energy, binding energy. Different docking methods. There are plenty of tools available, and these are the URLs for docking tools. So if you see this FlexX, Gold, Fred, Glide, Affinity, these are all commercial softwares. This is the software what we can able to try. Okay, these are the freeware. These two alone are the freewares. But trying this is more complicated. UCSF talk and the Autodoc is easier. So I'll be teaching you to how to use the Autodoc. The Glide is a product of the Scroid Inger. The Glide is a program, is a ligand protein docking of the Scroid Inger package. Like this, we have a Tripose. We have one package called Tripose. Even earlier in SVC, we procured this top, this package Tripose. The Tripose we can also use uh, this kind of a tool for docking. Now let me take you to this docking and dock. What is docking? Docking, I already told you, it's a binding, and we check through a confirmation. So confirmation, it means that if you see the confirmation, so this is one confirmation. One ligand is confirm is binding in this binding site. If you see in Discovery Studio, if it was the same, if the same ligand, if you try to dock in the Discovery Studio, and if you have 5 binding sites for the protein, the ligand can also bind with the other binding site. For example, the ligand can also bind in this binding site. If the ligand is binding in this binding site, this is actually a conformer too. Okay, so not just this is a conformer, even here, whatever you find here, this might be the best fit like a conformer one. And this kind of a benzene can have a flexible rotation. Instead of the benzene ring is facing this direction, the benzene ring can face even in this direction. So this is actually conformer too. Actually, you have a backbone of a lead compound where these fragments alone is going to be like a flexible, like a fragments are broken, and it detached and will be attached in different orientation, and whatever different orientation it have, you can call that as a conformers. So conformer one, conformer two, like this we can try to bind with as many number of conformers. In Autodoc, defaultly the setting it will be like to generate 10 base conformers. But the one what we get a negative maximum energy, that is the base conformer. Okay. So conformers, the ligand is binding in the site one, but with different orientations. The ligand alone will be like a flexible kind of a ligand. Is the functional groups detached and attached. Okay, and similarly, even in the binding site two, here also you have 1 to 10 conformers. So like this, we can have as many number of conformers. Okay, in specific binding sites of one at the same ligand with the same protein. Okay, now let me take you to this. Here they are giving a different definition. So, what is the definition they're providing? Identification of the low energy binding modes. That's what I told you. So, out of different conformers, the one which is going to give the maximum negative energy, that is the best conformer with the active side. So we are not just bothered about the conformation. We are also bothering about whether your ligand is going to our ligand is going to bind with the binding site of a receptor or not. If and if it binds with the receptor, then obviously drug dosage, number of dosages, everything will be leveraged. Okay, so this is most important, and even whether it's specifically targeting the disease or not, that depends on how effectively your ligand is binding in the binding sites of the receptor. Now we can see the steps in docking. Actually, we have can able to find 4 steps in docking: ligand preparation, site characterization, scoring grid calculation, and fourth is the docking. Prediction of the binding energy. I told you how the binding energy is predicted. The binding energy is predicted only through Gibbs-free energy. So change in Gibbs-free energy is the change of enthalpy to the difference of temperature and change in entropy. And you know that the change in Gibbs-free energy can also be calculated from final state and initial state. If this is your initial state before the reaction, before the binding, and if this is the final state after the binding, if the ligand is bound with the receptor, this is the final binding energy, and this can be calculated through this formula. If a reaction is going to happen, then you are going to use this R as universal gas constant, T as absolute temperature, Kd as the dissociation constant. You remember Kd is nothing but the inversely proportional to the association constant. So what is Kd here? Kd, if you see, if ligand is interacting with a receptor, whatever I told you that K1 is association, isn't it? So this is Ka. And ligand receptor, if it try to dissociate into ligand and receptor, this is what I call it as Kd. Association constant, dissociation constant. Got it? Of course, the Kd is the product. What is the product? Ligand concentration, receptor concentration to the ligand receptor concentration. This is what Kd. The formula of Kd is the product of ligand and receptor concentration to the ligand receptor concentration. Similar, Ka is just quite opposite to that of. So it's inversely proportional to the Kd. Got it? So this is something about the kinetics and the thermodynamics involved in molecular docking. Molecular docking. Still, what is molecular docking and what are the various applications? In biology or biotechnology, whatever we call it as molecular recognition is what molecular docking in computational. If you see here, antigen interacting with the antibody, what is this? Immunology, isn't it? Enzyme substrate binding, enzymology. Molecule transport, signal receptor. Can you give me an example for molecular transport? What kind of subject is this? Give me an example or else. What kind of a subject? Molecule transport. Any example? Hemoglobin. In hemoglobin, oxygen is going to transport through hemoglobin. Under that case, the hemoglobin will be oxyhemoglobin during the inhalation. So during the exhalation, CO2 is going to release, isn't it? We have another protein called carboxyhemoglobin. Actually, hemoglobin, coxyhemoglobin, carboxyhemoglobin differ with respect to the carrier molecule called oxygen and CO2. Obviously, even the structure varies with all these three proteins. A slight change in conformation can also varies at that time during the binding and unbinding. Okay, so the transport protein is also most important. I believe 1 or 2 days before one participant asked me, "Sir, where exactly we can find the significance of ions in molecular docking?" There are actually different mechanisms in binding of drug to the protein. One such is ion channel. Have you heard about the ion channel? The potassium and sodium ions, okay, can able to transport through the ion channel. Okay, so this is most important even for the drugs. Few drugs we take drugs in the form of ions, and only these drugs can able to transport the required kind of radicals or whatever it is through these ion channels. Okay, so I believe the participant anticipated this answer from me. Okay, but also I told you that during the docking, try to remove the heteroatom molecules like water molecules and do the docking, because the water molecules will hinder the ligand to bind with the appropriate binding sites. That's the reason I told you to remove the heteroatoms before docking a ligand with a receptor. Okay. Now, if you take this, the next concept is a signal transduction. Signal transduction. That is, this also you can find in immunology. Much of my PhD research involves in this signal transduction. For example, IL-2 ligand bind with the IL-2 receptor. GMCs of ligand bind with the GMCs of receptor. Tumor necrosis factor ligand binds with the tumor necrosis factor receptor. Like this, you see interleukins, tumor necrosis factor family, and most of the proteins, immune proteins, have a ligand and receptor. So these ligands bind with the corresponding receptors. So this gives you more kind of types of immunity. Okay, so this is of course a most important thing, signal transduction, and even for this, one should have a knowledge on molecular docking. Okay, so if you see in molecular docking, especially in pharma, okay, whatever we tell a ligand is going to bind with a receptor, we can call this ligand either as agonist or antagonist. Can you tell me the difference between the agonist and antagonist? Because I can able to see many participants in this course are from pharma background. Can someone tell me what is the difference between agonist and antagonist? Are you typing in the chat box? ATMET is another method. Okay. So I asked you a question like, what is the difference between agonist and antagonist? Can I mention this because we have less time? Agonist is a drug which is going to give a desired biological effect. For example, if I'm going want to treat a viral fever, and if my drug is going to treat viral fever, I call that as agonist. But whatever drug I take, if it gives an undesired effect, instead of treating the viral fever, it is going to give some the fungal contamination, fungal disease. Then we call that as antagonist. We anticipated a desired reaction, but we got an undesired biological effect. Okay. So, and then if you see this molecular talking methods, these were the theory. Actually, initially in 1890, the lock and key model was proposed by Fisher, and in 1958, Daniel Koshland proposed induced fit model. And this is what we are using in all the talking tools. From the Voyong, he established the conformation ensemble method, the lock and key. The ligand is generally a fixed kind of a conformer, and also receptor is also fixed. So rigid ligand binds with a rigid receptor. This is what we call it as rigid ligand, rigid receptor. This is what we call it as a lock and key. Okay, and the induced fit, if you see both the ligand and receptor before talking, this is before talking, this is after talking, undergo a kind of a conformational change. See, it looks like a cashew nut at this shape. And even this receptor is undergoing a conformational change to fit each other. Okay. So, this is what we call it as a inverse fit theory. Now, let me take you to the conformational ensemble. This is the third method. So, if you see the third method, okay, our ligand is going to bind with different conformers. I told you, no, that backbone of the ligand is fixed, but only the functional groups present on the surface of the ligand is going to attach and detach, and we get a different kind of a conformation. That is what we call it as a flexible ligand. The flexible ligand has a different conformations, like how we can able to rotate a hand in axial direction, in other kind of a directions, left and right, etc. Similarly, even the ligand with the functional groups can have all these kind of a directions. So whatever directions, whatever conformers we have, if you try to dock a ligand with a receptor, so this is what you can find the most negative energy. The maximum negative energy is in this pose. Okay, when compared to this. Okay, so then you can tell the 2 is the best conformer for the ligand, which is going to give a high negative maximum value. So binding ligand in different conformers is what we call it as a conformational ensemble method. So the best method what we use is the conformational ensemble in most of the tools. And if you see the docking can be classified as a small molecule nucleic acid. So even we have DNA inhibitors, isn't it? DNA topoisomerase inhibitors. So if you want to inhibit the DNA, inhibit the RNA, even we can try to talk the small molecule with that corresponding nucleic acids. We can also talk protein nucleic acids in case of DNA protein interactions, RNA protein interactions. So this can also be done in docking. And similarly, protein protein. Okay, so these are the types of dockings other than small molecule protein, whatever we're discussing from the beginning. We have different tools for doing this different docking. If you say in talking, we can able to see the chemical complementarity. So if you see the shape complementarity, like fitting the puzzles, okay, how you fix one puzzle with another puzzle, okay, this kind of fitting, we generally observe in pharmacophore. You remember? This is one of the drug design method. I told you, just fit a ligand with a receptor. Okay, that's also available in hybrid based screening. There is this comes under hybrid based approach. The pharmacophore can be a ligand based, or it can also be a hybrid based. I told you, no. So if it is hybrid based, the shape complementarity, where you can able to measure the shape complementarity through fitness value, fitness scores. Okay, but whereas in docking, we obviously observe the chemical complementarity. It means that if this is your ligand, which is having a hydrogen bond donor, and my receptor is going to have hydrogen bond acceptor on the surface, and if my ligand is going to bind with this binding site of the hydrogen bond acceptor, we can see only one bond is formed. We can able to see only one bond is formed. This is a kind of an aliphatic interaction. Aliphatic amino acid is interacting with the aliphatic kind of a ligand, okay, functional group of a ligand. But we also can able to see a chemical complementarity, that if my ligand is going to have a cyclic ring, and also if my amino acid is also going to have a cyclic ring. You remember the amino acid proline, okay, which is going to have a five-membered ring. Similarly, wherever you can find the ring in the amino acid, and if our amino acid is going to interact with the ligand rings, then obviously you can see, if you have six rings, you find six bonds are formed. If there are five rings, five bonds are formed. So if you see the binding energy, remember binding energy is what the scoring function. This is what determines how effectively a ligand is bound with the receptor. Okay. If binding affinity is determined through binding energy, the binding energy is dependent on multiple factors. The most important factor is number of hydrogen bonds. Because if a number of hydrogen bonds is more, obviously the attraction is more, and obviously the binding energy will be negative maximum. Okay. So and the number of hydrogen bonds, and also like this number of aromatic interactions, number of aromatic interactions, number of salt bleach formation, everything matters. For example, if my ligand is going to bind with four different amino acids, is going to bind with four different amino acids. The number of bonds, if you can see only the aliphatic bonds. So in four amino acids, you have find four hydrogen bonds. But if my ligand is going to have an aromatic ring and it is also going to the aromatic ring of an protein, then obviously if that is a six-membered ring, you can find the one interaction, one amino acid interaction itself, you can find six bonds or shared. Okay. So only through this only the binding energy varies. And we can also find in electrostatic two bonds. Hydrophobic is also a strong interaction with four bonds. Like this, you can see the number of bonds based on the bonds. You can see binding energy value varies. And whatever I told you, the negative maximum best conformer. See, this ligand is best bind with the receptor, like how a pen cap is going to fit with the pen. Okay, so if this is a best conformer, this kind of energy, whatever we calculate, is the global energy. Is the global energy. The other energy, whatever we have, this we call it as a local energy. We have multiple local energy, but we have only one global energy, which is negative maximum, which is negative maximum in value. So this is something about the talking. We have three types of talking. Only one type of talking is not possible based on the rigidity and flexibility of the ligand and receptor. The one which is not possible, flexible protein, rigid ligand. Flexible protein, rigid ligand, we can't talk. We don't have any tools developed for this. Initially, we have tools for this, but nowadays, whatever tools we get in the internet is a flexible protein and flexible ligand talking. Even in Autodoc, we talk like this only. So docking, see the poses. Did you see the animation? It is docking with different poses, the same ligand, same receptor, same binding site, but different conformers. With chemical complementarity, you can find the binding energy will be this. And if you see here, the binding sites prediction. How the binding sites are predicted? In Discovery Studio ligand fit, the ligand fit is used for docking a single ligand with a single receptor. This is the program in Discovery Studio. So to check the binding sites, to distinguish the binding sites of lipophilic and hydrophilic, we enclose the protein in a grid box and then we fill with water. Wherever the water is going to accumulate on the surface, the outer side, we call it as hydrophilic. If water is not going inside of the protein, then the buried site, we call it as hydrophobic binding site. Okay, this is how the binding sites are determined. Scoring methods. So we use the interaction energies between the ligand and receptor, desolvation and solvation energies, entropic factors. So all these things we use it for different scoring functions. I told you even in this discovery study, we have 8 different scores. We use a correlation of different scores. I believe I've circulated this case study one. So what exactly we talked with afrinase is a plant compound, talked with the target receptors responsible for cardiovascular diseases. This is again a case study too. Okay, this is actually a docking of a plant compound very known with two important target receptors of hepatocellular carcinoma. This is the 2D interaction image. Okay, now let us see how to work on Autodoc. Autodoc, actually we have to prepare initially a ligand file and protein file. Whatever we have these files, we have to convert this in the form of PDBQT. You remember? This I already explained you when I was providing a demo on PYRX. PYRX is used for docking multiple ligands with receptor. But whereas in Autodoc, we dock a single ligand with a receptor. And today I'll be providing you demo only to how to dock a single ligand with receptor. So docking analysis and MD simulation, molecular dynamic simulation, I'll be teaching you on tomorrow. So first, let us see clearly on the steps of autodock, how to dock a single ligand with a receptor. Okay. So after converting this ligand and PDF protein in the form of PDBQT files, later even we are going to create some intermediate files in the intermediate steps. Like we create a GPF. GPF stands for grid parameter file. Grid parameter file. That is, we enclose a protein in a grid. You remember the grid I already told you here. Okay, this is what we have a kind of a grid, and here you have multiple grid lines. You have multiple grid lines, like how you see a graph. In a graph, you have a major scale, minor scale. The major scale is just like a centimeter, and minor scale is a millimeter. Like this, a grid is actually is going to have a major scale and minor scale, and you can able to find a minor scale whatever I mentioned. Okay, whatever minor scale we are going to have. Okay, a small kind of a box. This box is actually a 1 angstrom in size. This is what I call it as a particle. If you want to see the particle, okay, if you want to see a particle of 1 angstrom, you can able to find in the grid box. Okay, you can able to find in the grid box. Now let me take you to the that slide. Okay. Okay, now if you see this grid parameter file, you can understand like this, and DPF stand for the doc parameter file. Okay, like after running the grid, we can run the doc. So in the docking, we can get the doc parameter file, and this grid log file is also generated while we run the grid. So now let me take you to the steps in Autodoc. Okay. So this is the general thing. Or we just start with this elegant and receptor file, only two files, but we create enormous intermediate files while we execute the major and minor steps. Okay, this is the final output. How a final output looks like. The final output looks like in the form of it generates in the form of energy. Binding energy here in this case, I showed you the best energy is minus 5.2, and this is the protocol. Okay, first prepare the protein file. We have these sub steps. Prepare the ligand. This is the second step, and I will be circulating this kind of a protocol in the WhatsApp after this session. Okay, so you can go through that. So first you should have this protocol, then only you can able to work clearly. The third is preparing the flexible residue file. Fourth is running auto grid calculation. Fifth is preparing the docking parameter file. Okay, so this alone will be working it today. Later analysis we can discuss it on tomorrow. So I have already created a folder called demo file in desktop. In the demo file, so after installing Autodoc, if you want to install the Autodoc, you have to install the following softwares, whatever I'm going to show it now. So to install the Autodoc, okay, we have to install four softwares in Autodoc. Okay, what are the four softwares? We have to install. I show you the Autodoc installation files. See, this is Autodoc Suite Windows, Autodoc Suite Windows, Chimera, EDU, PyMon, Python Molecular Weir. Okay, this is Python Molecular Weir software. Each and every tool are present in different home pages. These have different URLs to download. Autodoc Suite, Chimera, EDU, PyMon, MGL tools. MGL tools. So MGL tools and Chimera is in 32-bit version, but still we can install this in 64-bit version. These two tools we can use in both 32-bit version and 64. But if you have a 64-bit version, you have to install only 64-bit version of Autodoc Suite as well as Python Molecular Weir. These two softwares alone we have to install in 64-bit version. Okay, EDU PyMol 64-bit version and Chimera in 64-bit version. Sorry, Chimera in 64-bit version. But NGL tools and Autodoc suite, whatever available in 32-bit version, you can download and you can install in any random fashion. It clear you won't get any problems in installation. Okay, there won't be any compatible problems. It successfully installed. After the successful installation of the Autodoc, you can find these icons. This blue icon is the Autodoc tool, and this also CADD. You can get this second icon like this. Third icon is Python molecular wear PMV, and the fourth icon is the Vision 1.5.6. These are the five icons you can get after installing these five softwares. Then if you want to open the Autodoc software suite, double click and open that blue color icon. If I double click and open the blue color icon, we can see this Autodoc 1.5.6 is going to get opened. And this is what we call it as Autodoc 4.0. This is what we call it as Autodoc 4.0. So if you install this softwares, okay, in the program files, you can find Autodoc 4.0. We can able to find Autodoc 4.0, Autograde 4.0 exe files. So I suggest you to copy the Autodoc 4.0 and Autograde 4 exe files in a folder, okay, in a separate folder where you're going to work. And I'm going to save even this Autodoc tutorial in the same folder. And the protein file, whatever protein and receptor, whatever we're going to dock, that also you're going to hold in the same folder. So now let me take you to this. I already pasted a protein file. The protease protein is from HIV. Okay, so for a HIV protease, I'm going to dock with my ligands, and I'm going to check which is best bind with the HIV protease. So I have chosen this kind of a protein. Actually, this protein is very small in size. It's very small in size, and you have only single chain. You don't have heteroatoms. If you have heteroatoms and if you have multiple chains, and each and every chain has the same kind of an amino acids in a sequence, then I suggest you to use a single chain. Use a single chain, delete the other chains, and also remove the heteroatom molecules. First, prepare a protein, then you can try it for docking. So this protein is already prepared. So I have this protein. Now I'm going to take one ligand. For taking a ligand, I'm going to take from NCBA PubChem. So I'm going to check this HIV protease. Think like it's a normal protease protein. Okay, for a protease, I'm going to check the binding affinity with one compound. What compound can I try? Can I try with aspirin or some kind of a plant compound? Can I aspirin is something different. So I can try with some compound. Uh, can be a pyrovinol. Okay, some pyrovinol B. I'm going to try with a compound called pyrovinol B. This is a rotenide compound. Okay, this compound I'm going to download. STF. So in desktop, I'm going to save it in the same folder. The ligand I kept as a pyrovinol B. I saved it, but in the form of STF. Just because this compound is in the form of STF, I am going to convert this STF format into PDB format initially. To convert the STF format into PDB format, because PDB format is not available in PubChem. So my objective is to first initially create this compound in PDB. So I right click that compound and I opened it. Now we are seeing this compound. I'm going to just save this compound as PDB. So pyrovinol B in PDB file, I'm going to create. Okay, pyrovinol B dot PDB. I am going to save it now. Now initially we had STF. Now I converted as PDB file. So you can see this is the recently saved PDB file. Okay, so we can see now the ligand is there, protein is there. Only thing is that we have to step on. So for stepping in, so first go to the tutorial. So I already showed you the major steps. Let me walk one by one. Okay, so first I have to open the Autodoc tool. Where is my Autodoc? Autodoc is not yet open because the Autodoc is Of course, whatever Autodoc I open is a commercial software. It works only, I think, 30 days trial, I believe. So, only thing is that we have to click remind me later. If I click remind me later, it can be able to use still the free version of Autodoc. The first step, what you have to make is set the path of the ligand and receptor. For this, go to the file preferences setup. And inside that, you have a startup directory. In the startup directory, locate the directory where you find the receptor and ligand. I just that is demo file. I just typed as demo file. Now click make default set. Okay, whatever location you have the ligand and receptor, and now I have set. Now after setting it in startup directory, click dismiss. So the first path, the default path, but which is not mentioned in this tutorial, that alone I didn't mention. You just follow that step to locate the ligand and receptor in one single path. Now we are going to take steps. The first step is file read molecule. File read molecule. Okay, and select protein name. Can I go across that? File read molecule. The molecule what I have to read is the macromolecule. So I'm going to open the protease. So in that location, you can able to see the protein and ligand, but I want to open protein. I opened it. The next step is to go with the adding hydrogens and charges. First, they ask you to add hydrogens, then charges. For this, use editor. Okay, use hydrogens, add. Okay, all hydrogens is in default. Whatever parameters in default, you keep it and click OK. See the confirmation change. Earlier, we don't have white color. The white color, whatever we are seeing here, is now hydrogens. The hydrogens are added. Now, next, edit, add Coleman charges. Edit, charges, go to the add Coleman charges. The Coleman charges added, simply click OK. The next is file save write PDB OK. So while saving it, file save write PDB, it gives you this kind of a window. Defaultly it is in demo file, but I'm going to save it as protein1, protein1.pdb. Earlier it is protease.pdb. After adding hydrogens and charges, I renaming it as protein1.pdb. This is how I'm going to save the file. For a demo, you can try to use however I'm using. Now let me take you to the preparing the ligand. For preparing the ligand, go to the ligand input open ligand. So ligand input open ligand. So this is what you are going to click. So according to this step, they're asking you to open the ligand, but unfortunately we don't find any ligand. So why it's not there? Because default is asking in PDBQT. Whether we have saved the ligand in PDBQT? No. Recently, we have saved that HDF as PDB. Just because it's not present in PDBQT, you have to select all files. Then only we can see this pyrimidine B in PDB format. Whatever we have pyrimidine B in PDB format, click open. So after clicking open, you can see that the ligand is opened, and the other parameters like gas sticker charges, matching the hydrogens, aromatic carbons, all these things you can find. Now our ligand is here, receptor is here. Still, we didn't taken steps for docking. We just placed in this kind of a visualizer window, Python molecular visualizer window. So after ligand input, then the next step is ligand torsion tree detect route. Ligand torsion tree detect route. So this step is completed. No signs you can able to see here. Ligand output save as PDBQT. Whatever we have done, ligand output save as PDBQT. Here also I suggest you to use something like ligand1.pdbqt. Like however we have protein1.pdbqt, now I'm going to have ligand1.pdbqt. So this is how I've saved it. Okay, so whatever I completed is up to this step. That second step is completed. The next step is the third step. The sub step is grid macromolecule choose. Or you can, wherever you find choose, you can also use an option called open. Okay, grid macromolecule open, select protein molecule. Grid macromolecule open. So if you click open, you can able to see whether protein is present or not. Only ligand is recently saved as PDBQT. Till now, we didn't save protein as PDBQT. The latest thing what we have saved is protein1.pdb. So just because we have saved it as protein1.pdb, it is present in only PDB format. So click all files. Now we can find the recently saved protein which is in PDB file format. So select this protein one, click open. So whatever I opened, so it's asking you to kind of a warning. Nothing, don't worry, don't get panic. Simply it's asking you once again to save like PDBQT format. So it's automatically we got this kind of a pop-up box. So now we have to type like PDBQT. So this protein one earlier we had PDB format. Now I am saving it as PDBQT. So protein one.pdbqt save. So I have completed up to this step, a part of third step. The next is to define this binding sites or active sites. For this, select from string, and then you have to go with select protein residues active sites. Can I go with this? Select from string. In this, we have molecule list, isn't it? Click this molecule. Our recently saved protein is protein 1. So that only you have to select here. And if you want to use all these atoms, if the binding site can be any of these atoms of this protein. So if you want to select all the atoms of that particular protein, click asterisk. So I mean type asterisk. In atom, you have to type asterisk. Okay. And then you have to simply click only once. There is a button called add, click once. If you click twice, you won't get the result. Be careful in executing this step. Then click dismiss. Only once you have to click that. Now this step is completed. Now we are moving to the fourth step, running autogrid calculation. So create setmap types, open ligand. Create setmap types, open ligand, open the ligand. The ligand is ligand1.pdbqt. So this step is completed. Wherever you find choose, click open. So I've completed up to this step now. The next is grid box. So grid box. The grid box is very small. If I just drag and show you, you can see you can find a cubicle box, but this cubicle box is not perfectly enclosing all the entire protein. Our objective is to enclose the entire protein. So for this, right click and you first click apply so that everything gets activated. Now, whatever we have maximum of 126, I give the same value 126. Click OK. Just because it's a small protein, even in the small coordinates, you can get uh, the protein is going to get encapsulated in this grid. 126. Yes, this protein molecule is surrounded by the grid in all the directions. Okay, the blue alone is small. You can increase the size of the blue to slightly 135 as the maximum, and this also 135 as the maximum. Okay, so I have perfectly fit my protein in this grid box in all the directions. So click File, close saving current. So now I won't miss any binding sites if I try to dock ligand with the receptor because I bound with the grid. So up to this I have completed. The next is grid output save file as GPF. Grid, okay, output save GPF. Now we are going to save it like grid1.gpf. I've saved it. The next step is to go for run autograde. Run autograde. So in autograde 4, you have to select the autograde 4.exe file, and in grid1.gpf, you select this grid1.gpf file. So also you can find this is loaded automatically. Grid log file is loaded, and even this entire URL is automatically filled. If it is filled, then simply click launch. It takes some time to generate the results if you bind ligand with the receptor. If your laptop is having a good configuration, okay, which is having a good processing capacity and having a good RAM capacity, even this running the autogrid, which takes more time when compared to any other steps in this, can also be completed as soon as possible. So we have to wait for the next step until this white window getting closed. If the white window immediately closed, then you have made some mistake in the earlier steps. Just because this white window didn't close immediately, but after some time, after a few seconds, it's closing, it means that we have successfully executed that fourth step. Now we are moving to the fifth step. We just complete 5 and 6 steps within 2 or 3 minutes. I mean, I will be completing the session within 2 to 3 minutes. The next step is going with the docking macromolecule set rigid file. Docking macromolecule set rigid file. So macromolecule, you have to select only macromolecule. So okay, that's what it understand. So click open. So we have completed this step. What step? This step completed. Docking ligand, choose ligand. Okay, docking ligand, instead of choose, better find open. Okay, click that ligand one.pdbqt because they're asking to ligand open. So I have opened the ligand. So default you get this kind. Click accept. So this step is completed. Docking search parameter genetic algorithm. Docking search parameter genetic algorithm. So default you find 10 poses, 10 conformers. If you try to dock a ligand with 10 conformers, it takes more time to generate the result. If I want to get the best two conformers, type 2. In number of genetic algorithm runs, I type 2 and click accept. So I'll be getting the best two poses. Now, docking output American genetic algorithm. Docking output American genetic algorithm. Now I'm going to save this file as doc1.tpf. Doc1.tpf docking parameter file. This is completed. Next, run autodoc launch. This is the last step. Run autodoc and select the autodoc 4, and here you have to select the doc1.tpf. So all the URLs you can able to find it. Filling. Now click launch. Now click launch. Okay. Then obviously you can able to see you are running this. Oh, the window not yet closed. Yeah, okay. So if the window getting closed, then we have to execute the sixth step. The sixth step is analyze dockings open, analyze macromolecules open, and then we complete these steps. Can we see whether this white window is closed or not? Yes, now the white window is not yet closed. Yes, now I think it's getting closed. Not. So after this, we have to execute only 3 to 4 steps. If you close in between, that is after closing the white window or even between the, if you close in between, then you have to execute all the steps from beginning. So now only the white window got closed. So now I'm going to execute the sixth step. Analyze dockings open. Analyze dockings open. So we have to open this doc1.tlg. So defaultly whatever you get, click OK. And now again click OK. Then analyze macromolecule, choose. That is analyze macromolecule, instead of choose, click open. Nothing get happen. You just go to the next step. Analyze conformations load. Analyze conformations load. So you'll be getting, this is the answer. So this is the answer we are anticipating. So we got our Beryllium B is docking with a protease with the binding energy is minus 7.65 kilocalorie per mole. It means that it gives a significant kind of a result. If it is minus 6 and below, then we can tell our ligand is best binding with the receptor. Then the next step is analyze conformations, play rank by energy. Analyze conformations, play rank by energy. Now we'll be getting like this ampersand sign in this window. Click this ampersand. Now click build current. Then click write complex. So while writing the complex, instead of PDBQT, convert it as all. So our ligand is actually BB. So I better to use the double quotes BB underscore protease underscore complex. So I talked Pyronol B with a protease, and this I'll be saving it like BB protease complex dot PDB. So whatever I am going to save, I enclose it in double quotes. That's it. This is the completion of docking. I got the score, and to analyze the docking score results, we'll be discussing it on tomorrow. Okay. Now we can safely close all these windows. So the results, the intermediate files are already available in that folder. So you can able to open and study at any time. But if you close the window in between, you can't able to execute once again. And you have to start from the beginning. So I have completed my today's session. Okay. Any doubts, you can ask me at present. Any doubts? Thank you, madam, for appreciating. If you have any doubts, you can ask me, or else we can move on tomorrow for the next session. Okay, have a great day to you all. Bye.